Welcome to the Quiet Lab, a calm space where science slows down and your mind can rest. If you enjoy learning while you unwind, consider subscribing. It helps support future episodes and gentle explorations like this one. Let us know which part of the cell's journey you found most interesting or surprising. Everyone connects with something different. Tonight, we'll uncover a hidden world, so tiny it's invisible without a microscope. And yet, without it, life wouldn't exist. We're talking about the cell, the fundamental unit of all living things. Some organisms are made of just one. Others, like humans, are made of trillions. But whether simple or complex, every form of life begins with this tiny building block. It's a story of structure, purpose, and the quiet machinery of life. Let's begin. A cell is the smallest structure that can perform all the basic functions of life, like getting energy, responding to its surroundings, and reproducing. And while cells may seem tiny and uniform, they're surprisingly diverse. Structurally, there are two main types of cells, prokaryotic and eukaryotic. Prokaryotic cells include bacteria and archaea, simpler cells that lack a nucleus. Eukaryotic cells, found in plants, animals, fungi, and protists, are more complex and contain a nucleus, along with other specialized compartments called organelles. Now that we've seen the broad categories, let's explore each one a little more closely. Starting with the prokaryotes. These are the simpler kind of cells, but simple doesn't mean unimportant. In fact, prokaryotes were the first forms of life on Earth, ancient organisms that shape the very foundation of biology as we know it. Bacteria are the most well-known examples, found in soil, water, and even inside the human body. But there's also archaea, a lesser-known group that often lives in extreme environments like hot springs, salt lakes, or deep-sea vents. Though they resemble bacteria under a microscope, archaea have distinct molecular structures that set them apart, including differences in their cell membranes and how they process genetic information. Prokaryotic cells don't have a nucleus. Instead, their genetic material floats freely within the cell, often in a single circular chromosome. They also lack the membrane-bound organelles we see in more complex cells, like mitochondria or the Golgi apparatus. Still, prokaryotes are remarkably efficient. They recycle nutrients, support ecosystems, and even help our own bodies function, aiding digestion, producing vitamins, and protecting us from harmful microbes. Though small and structurally simple, their role in the web of life is enormous. Without them, life as we know it wouldn't be possible. Now, let's talk about eukaryotic cells, the kind found in plants, animals, fungi, and protists. These cells are more complex than their prokaryotic counterparts, with a clear internal structure and well-defined compartments. At the heart of each eukaryotic cell is the nucleus, a membrane-bound center that safely houses the cell's genetic material. It's like a control room where instructions for the cell's activities are stored, organized, and protected from outside interference. Surrounding the nucleus is a dynamic space filled with organelles, specialized structures that each perform essential tasks. Some generate energy, others build proteins. Some transport materials or break down waste to keep the cell balanced and clean. This internal layout is one of the key features that sets eukaryotic cells apart. By separating different functions into distinct areas, the cell can perform multiple processes at once, more efficiently and with greater control. The cells we often study under a microscope, like cheek cells, onion skin, Elodea leaves, or paramecium, are all examples of eukaryotic life. And while they come in many forms, they share the same basic internal structure: a nucleus, a flexible membrane, and a coordinated system of organelles. Now, let's compare two major types of eukaryotic cells, animal cells and plant cells. We'll start with the kind you're made of. These are the cells that make up your body. including the ones that line your cheeks, skin, muscles, and brain. They're typically soft and flexible, without a rigid outer wall, which allows them to change shape slightly when needed. The outermost layer is called the cell membrane, a thin, protective boundary that surrounds the cell and helps regulate what comes in and out. It also plays a role in communication, using embedded proteins to sense signals from neighboring cells or the external environment. Animal cells don't contain structures like chloroplasts since they don't perform photosynthesis. Still, they're incredibly busy, constantly producing proteins, generating energy, and sending signals to other cells to coordinate movement, healing, and internal balance. Plant cells, like those found in onions, leaves, or Elodea, have some extra structures that set them apart. The most noticeable difference is their shape. Plant cells often appear more rectangular, thanks to a sturdy structure called the cell wall. The cell wall surrounds the cell membrane and gives the cell its shape, strength, and protection. Made primarily of cellulose, a tough, thread-like material, the wall helps plants stand upright and resist external pressure. Just inside that wall is the cell membrane, which still plays a crucial role in controlling what enters and exits the cell. Another key feature of plant cells is the presence of chloroplasts. These green, rounded organelles capture sunlight and convert it into food through a process called photosynthesis, something animal cells never do. Chloroplasts contain the pigment chlorophyll, which gives plants their green color and helps absorb light efficiently. Despite their differences, both plant and animal cells share many important structures. Let's look closely at the first one, the cell membrane. The cell membrane is like the gatekeeper of the cell. It surrounds the entire cell in animals, and in plants, it sits just beneath the cell wall. This membrane isn't a solid wall. Instead, it's made of two layers of molecules called phospholipids. Each phospholipid has a hydrophilic head, meaning it likes water, and a hydrophobic tail, which repels water. When arranged together, they form what's called a phospholipid bilayer. This structure creates a flexible barrier, one that lets some things in and keeps others out. Scattered throughout the membrane are proteins that act like gates, channels, or sensors. These proteins help the cell take in nutrients, release waste, and respond to signals from the outside world. This selective control is what makes the membrane selectively permeable. It carefully chooses what to allow through. This is key for maintaining the cell's homeostasis, its internal balance. Once you pass through the membrane, you enter a substance called the cytoplasm. It's not empty space, but a thick, jelly-like fluid filled with nutrients, enzymes, and structural proteins. The cytoplasm fills the area between the organelles, holding everything in place and allowing chemical reactions to take place smoothly. If you were to zoom in, you'd see molecules constantly moving, enzymes breaking down nutrients, and messages being sent from one organelle to another, all within this seemingly quiet gel. The cytoplasm is alive with activity. At the center of most eukaryotic cells is the nucleus, often called the command center. It's surrounded by a nuclear envelope, a double membrane lined with tiny pores. These pores carefully control what enters and exits, keeping the genetic material protected while allowing instructions to pass through. Inside, the nucleus holds a cell's DNA, its complete genetic blueprint. At this stage, DNA isn't tightly packed into chromosomes. Instead, it exists as long, loose strands called chromatin, a relaxed form of DNA that stays easily accessible when the cell is not dividing. This DNA carries the instructions for everything the cell does, from which proteins to build to when and how to divide. Also housed within the nucleus is a smaller, dense structure called the nucleolus. The nucleolus builds ribosomes, the tiny molecular machines that will later assemble proteins from the DNA's instructions. This process of following DNA's instructions begins inside the nucleus, with a step called transcription. Transcription is how the cell makes a working copy of a specific gene. Think of DNA as the master blueprint, and mRNA or messenger RNA as a portable version of just one instruction page. During transcription, a short segment of DNA is copied into a strand of mRNA, a molecule designed to deliver the instructions. Once completed, the mRNA leaves the safety of the nucleus through tiny pores in the nuclear envelope and enters the cytoplasm. But the message isn't useful on its own. It still needs to be read and translated into something the cell can use, a protein. To do that, the mRNA must reach its next destination, a tiny but powerful structure perfectly designed to read its code and carry out its instructions. That destination is the ribosome, the starting point of protein synthesis and one of the most essential machines in the cell. It is the cell's protein factories. They're made up of two parts, a large subunit and a small subunit that come together like a clamshell to hold the mRNA steady as it's read. Some ribosomes float freely in the cytoplasm, building proteins that will be used right there inside the cell. Others are attached to a special organelle called the rough endoplasmic reticulum, which we'll get to in a moment. For now, let's follow the process of translation, the step where mRNA is decoded and proteins are built. The ribosome reads the mRNA 3 bases at a time. Each group of 3 letters is called a codon. Each codon tells the ribosome which amino acid to add next. Amino acids are the building blocks of proteins, like beads on a string. One by one, the ribosome links these amino acids together into a long chain. As the chain grows, it starts to fold and curves into just the right shape, becoming a functional protein. Some proteins are enzymes. Others are hormones. Some build structures. Others send signals. The shape of each protein determines what it can do, and every protein starts its life here, at the ribosome. From here, we move on to the rough endoplasmic reticulum, often called the rough ER. This structure is like a network of folded membranes that surrounds the nucleus, a bit like an internal highway system. It's called rough because it's covered in ribosomes, giving it a bumpy appearance under a microscope. Proteins made by these ribosomes are usually destined to be shipped somewhere, either to other organelles, to the cell membrane, or even out of the cell entirely. Once a protein is built by a ribosome on the rough endoplasmic reticulum, it slips inside the ER's folded spaces. There, it begins to fold more completely into its functional shape. Sometimes the protein is modified. A sugar group might be added, or a small molecule attached. These modifications are like finishing touches, fine-tuning the protein for its job. Once the protein is finished in the rough ER, it needs to move on to the next station in the cell's delivery system. That station is the Golgi apparatus. The Golgi apparatus, also called the Golgi body, is one of the most visually striking organelles. It looks like a stack of pancakes, gently folded and layered. Its job is crucial: to receive, modify, and ship proteins. When a protein leaves the rough ER, it's enclosed in a small bubble called a vesicle, like a delivery box. The vesicle carries the protein to the Golgi, where it merges with the Golgi's membrane. Inside the Golgi, the protein is further processed. The Golgi might add a carbohydrate group, attach a lipid, or help the protein fold even more precisely. Once the protein is ready, the Golgi packages it again into a new vesicle. Now, the vesicle knows where to go. It might deliver the protein to another part of the cell membrane, where it can be released, or it may even be sent outside the cell entirely. The Golgi apparatus ensures each protein is correctly tagged, folded, and addressed. It's the final checkpoint before delivery, and without it, the cell's internal logistics would fall apart. But once everything is sorted and ready, how does it actually move? The cell needs a transport system, something to carry these molecules safely to their destinations. That's where vesicles come in. These small spherical sacs are made of the same kind of membrane that surrounds organelles. They pinch off from places like the Golgi and the endoplasmic reticulum, float through the cytoplasm, and fuse with their target, delivering their contents with precision. Now let's pause and trace the journey so far. DNA in the nucleus holds the instructions. A segment is copied into mRNA, transcription. The mRNA leaves the nucleus and is read by ribosomes, translation. Ribosomes build a protein using amino acids. If the ribosome is on the rough endoplasmic reticulum, the protein enters the endoplasmic reticulum for folding and modification. It travels in a vesicle to the Golgi apparatus. The Golgi modifies and packages the protein. A final vesicle carries the protein to its destination, inside or outside the cell. This whole process happens constantly in every cell of your body. Right now, millions of proteins are being built, folded, shipped, and used just to keep you alive and well. So, we've seen how proteins are built, modified, and shipped, a carefully coordinated process involving the nucleus, ribosomes, the rough ER, and the Golgi apparatus. But there's much more happening inside the cell. Beyond protein production, there are structures that manage energy, detoxify substances, and recycle worn-out parts, all working together to keep the cell alive and in balance. Let's continue our exploration with a few of these essential players: the smooth endoplasmic reticulum, lysosomes, and the mitochondria, the cell's powerhouse. Let's start with the smooth endoplasmic reticulum, often called the smooth ER. If the rough ER is like a protein assembly line, then the smooth ER is more like a detox center and chemical lab. The smooth endoplasmic reticulum doesn't have ribosomes on its surface. That's why it looks smooth under the microscope. It's made of the same folded membrane system as the rough endoplasmic reticulum, but its jobs are different. The smooth ER is responsible for making lipids, especially phospholipids for membranes and steroids like hormones, detoxifying harmful substances like drugs, alcohol, or toxins, storing calcium ions, which are important for muscle contractions and cell signaling. In liver cells, the smooth ER is especially active, helping to clean the bloodstream and break down chemical byproducts. And in cells that produce hormones, like those in the adrenal glands, the smooth ER is busy making steroids that help regulate your body's functions. Though less famous than rough ER, the smooth ER plays a key role in keeping the cell chemically balanced. Next, let's meet the lysosomes, the cell's recycling and digestion centers. Lysosomes are small, membrane-bound sacs filled with enzymes. Their job? To break things down. That includes worn-out organelles, misfolded proteins, invading bacteria or viruses, and even large food particles taken in by the cell. You can think of lysosomes as acidic bubbles that dissolve unwanted or damaged materials, reducing them to simpler parts that can be reused or expelled. The enzymes inside a lysosome work best in an acidic environment, with a pH of around 5, and stay safely enclosed until needed. This containment is important. If they were to rupture, the powerful enzymes inside could damage the entire cell. That's why they're so carefully managed. In developing organisms, lysosomes also play a role in programmed cell death or apoptosis, a way for the body to remove cells that are no longer needed. It may sound harsh, but it's a vital part of growth and healing. From digesting bacteria to recycling old parts, lysosomes keep the cell clean, efficient, and protected. Let's turn to one of the most well-known organelles, the mitochondrion, often called the powerhouse of the cell. Each mitochondrion is like a mini power plant. Its job is to convert the energy stored in food, like glucose, into a usable form of energy called ATP. ATP stands for adenosine triphosphate, and it's the energy currency of the cell. Without it, nothing would move, build, or function. Inside each mitochondrion are two membranes, an outer one and a highly folded inner one. These folds are called cristae, and they give the mitochondrion more surface area to perform its reactions. The process of turning glucose into ATP is called cellular respiration, a multi-step chemical process that uses oxygen to extract energy from sugar molecules. This is why your body needs oxygen, not just for your lungs, but for your cells. Here's how it works in simplified terms. Glucose from food enters the cell. Inside the mitochondrion, it goes through a series of reactions. Oxygen is used to help break the glucose apart. The energy released is captured in molecules of ATP. Each glucose molecule can produce between 36 and 38 ATP molecules, depending on efficiency. That's a lot of energy, enough to power all the processes your cell needs to live and grow. Here's a fascinating fact. Mitochondria have their own DNA. Unlike other organelles, mitochondria carry a small loop of genetic material that's separate from the cell's main DNA in the nucleus. This mitochondrial DNA is passed down from mother to child, and it contains instructions for a few proteins that the mitochondrion needs to function. Because of this, scientists believe mitochondria were once independent bacteria that formed a symbiotic relationship with early cells, a theory known as the endosymbiotic theory. It's a beautiful example of cooperation at the microscopic level, a tiny partnership that gave rise to complex life. All across the cell, everything is in motion. Lipids are being built, proteins are folded and shipped, energy is transformed, waste is cleared. It's not chaos, it's coordination. Each organelle, though different in function, plays a role in the larger system. Like instruments in a quiet orchestra, together they maintain balance, repair damage, and power the processes that sustain life. This work happens all the time, quietly, in the background, and it's happening inside you right now, in every part of your body. And yet, for all their importance, these organelles are invisible to the naked eye. So let's change that. Let's step outside the inner machinery and take a look through the microscope at real cells. We'll observe cheek cells, onion cells, Elodea, and paramecium, and see what they reveal about the diversity of life, even at the cellular level. Under the microscope, a quiet world comes into focus. To prepare cells for observation, professionals often use biological stains, specialized dyes that help highlight specific cell structures under the microscope. Methylene blue, for instance, is commonly used to make nuclei in animal cells more visible. Iodine is another example. It helps reveal the internal features of plant cells, such as those in onion skin. These stains are typically used in both classroom and laboratory settings to improve contrast and clarity. In some cases, they may alter the cell or affect living function, which is why certain stains are chosen specifically for live or fixed samples, depending on the goal of the observation. Whether the sample is alive or preserved, stained or unstained, the structures viewed under the lens offer a meaningful glimpse into function, organization, and adaptation. These are cells, real ones, living or once living structures, each with its own shape, texture, and purpose. Let's take a closer look at a few of them and see what they reveal about the diversity of life, even at the cellular level. Cheek cells are one of the most common human cells viewed under a microscope, and one of the easiest to recognize. Collected with a simple cotton swab, they offer a clear view into our own biology. What you'll see is a soft, irregular shape, not sharp-edged or stiff like a plant cell. That's because cheek cells don't have a rigid wall. They bend, shift, and move with the tissue around them. Along the edge, the cell membrane forms a delicate outline, holding everything together. Inside, the nucleus is usually the most visible feature, a darker, rounded structure where the DNA lives. The cytoplasm around it can appear slightly grainy or translucent, depending on the lighting and stain used. They may seem ordinary, but these cells are constantly working, forming a barrier, helping with absorption and replacing themselves as needed. Even here, in a part of the body we rarely think about, cells are living out the same quiet cycles of growth, function, and renewal. But not all cells look or behave the same. Let's turn to a plant cell next, one you've likely encountered in your own kitchen. You've probably chopped an onion before, but you've never seen one like this. Just beneath the outer layers is a thin, nearly invisible sheet, the onion's epidermis. When placed on a slide and magnified, a hidden pattern appears. Rows of long rectangular cells, clear, consistent, and neatly aligned like tiles. That distinctive shape comes from the cell wall, a rigid outer layer that helps the plant hold its form. You may spot a nucleus slightly off center, or see how the cytoplasm gently hugs the inside of the wall. What you won't see are chloroplasts, and that's not a mistake. Because onions grow underground, they don't need to absorb sunlight, and so their cells never develop those green, light-harvesting structures. Even without color, onion cells are a perfect example of how organized and uniform plant tissue can be. A quiet order hidden inside something we use every day. In contrast, some plant cells show more than structure. They reveal activity in real time. Elodea is a freshwater plant, often found in aquariums, but under the microscope, its leaves reveal something far more dynamic. The cells are shaped like rectangles, stacked tightly together, but what makes them stand out is what's happening inside. Dozens of chloroplasts, small green structures, drift through the cytoplasm in looping paths. This movement is called cytoplasmic streaming, and it's more than just motion. It helps distribute nutrients and keeps the cell functioning efficiently. If you watch closely, it feels almost like traffic, quiet, purposeful, and steady. You might also notice the central vacuole, taking up most of the space inside each cell. It stores water, helps with structure, and maintains internal pressure so the plant stays upright. Elodea is one of the few plant cells we can observe alive, in motion, and at work, a rare chance to see life unfolding in real time. Now, let's shift again, this time to a protist. Protists are not plants, not animals, not fungi. They're in their own category, single-celled organisms that do it all. Paramecium is one of the most famous. Under the microscope, it looks like a slipper-shaped creature, gliding smoothly through water. Its outer edge is covered in fine, hair-like projections called cilia. Paramecium is not part of a tissue or an organ. It is an entire organism, in just one cell. Inside, you might see two nuclei, a large one for everyday function and a small one used during reproduction. You might also observe food vacuoles, tiny bubbles where it digests what it consumes, and a contractile vacuole, which squeezes out excess water, keeping the cell from bursting. Paramecium shows us that even a single cell can swim, eat, sense, and survive, all on its own. It's a reminder that life doesn't need to be large to be complete. Now that we've seen what various cells look like under the microscope, let's take a closer look at a few key features. Structures that stand out in plant cells and play important roles in survival. In plant cells, like Elodea and onion, one structure stands out, the central vacuole. This is a large, water-filled sac that can take up 90% or more of a plant cell's volume. Its main job is to store water, but it also holds nutrients, waste, and sometimes pigments. The vacuole creates pressure against the cell wall, helping the plant maintain its shape. This pressure is called turgor pressure. If the vacuole loses water, for example during a drought, the cell shrinks inward and the plant wilts. This shrinking is known as plasmolysis. Though invisible to the naked eye, the vacuole is a powerful force in plant survival. While the vacuole helps the plant stay upright and hydrated, other structures add something more colorful. Signals that speak not through structure, but through appearance. Some plant cells don't contain chloroplasts. Instead, they carry pigments of different colors. These are stored in chromoplasts, organelles that hold red, orange, or yellow pigments. You'll find chromoplasts in flowers, fruits, and vegetables like carrots and tomatoes. Unlike chloroplasts, they don't carry out photosynthesis, but they attract pollinators and animals that spread seeds. Their color is a signal, an invitation, and a reflection of the cell's purpose in the larger world. Finally, let's visit the underside of a leaf. If you examine a prepared slide or a fresh sample, you might notice small openings, each surrounded by two curved cells. These are stomata, tiny pores that allow the plant to breathe. Each stoma is flanked by guard cells, which swell to open the pore when water is abundant and shrink to close it when the plant is dry. Through stomata, plants take in carbon dioxide and release oxygen and water vapor. This delicate balance of gases is essential for photosynthesis and for life on Earth. They're almost invisible, but each one plays a part in the air we breathe. Let's return to the microscope one last time, not to discover something new, but to reflect on what we've already seen. Cheek cells, onion layers, Elodea leaves, a paramecium drifting through a drop of pond water. They may look different, and they are, but what connects them is more fundamental than what separates them. Each one is built from the same components: a cell membrane to separate inside from out, cytoplasm to hold everything together, DNA to provide instructions, organelles to carry out life's work. And in each case, those parts interact in a structured, purposeful way. Cells don't move randomly. They aren't passive. They are neat systems, managing energy, responding to changes, building molecules, recycling waste, and transmitting information. And whether a cell is part of a leaf, a cheek, or a free-living protist, its function depends on the precise work of these internal structures. Let's step back for a moment. Across all of biology, there are a few ideas that keep reappearing, patterns that reveal how life is both diverse and unified. Cells are one of the most important examples. All living things are made of cells. All cells come from pre-existing cells. These two principles form the core of what's known as the cell theory, a foundation of modern biology. It doesn't matter whether you're looking at a multicellular plant or a tiny single-celled organism. Every living thing depends on the activity of its cells. What's remarkable is how cells do their jobs in similar ways, even in very different types of life. For example, cheek cells and a paramecium both have nuclei, even though one is part of a human body and the other lives on its own as a microscopic organism. Onion cells in certain aquatic plants use large central storage sacs for support and structure. All eukaryotic cells use ribosomes to build proteins, energy centers to generate power, and membranes to control their internal conditions. These shared features allow scientists to study one kind of cell and apply that knowledge to others. Understanding how energy is made in yeast can help us understand how it's made in muscle cells. Studying how light is turned into food in green plant cells helps explain how plants survive. There are differences, of course, parts that allow cells to take on special roles. Plant cells have firm outer walls and green parts for capturing sunlight. Animal cells don't have those, but may include small helper structures involved in cell division. Some tiny single-celled organisms have unique features, like tiny hairs for movement or small sacs that help manage water. But beneath these differences is a shared logic, a common set of rules that govern how cells operate. Cells are efficient because they organize their tasks into different sections inside the cell. This is especially true in eukaryotes, where membrane-bound organelles allow different processes to happen in parallel. The nucleus protects genetic information. The endoplasmic reticulum handles protein folding and lipid production. The Golgi apparatus sorts and modifies products. Lysosomes manage digestion and recycling. Each organelle contributes to the cell's larger goal, to maintain homeostasis and support the organism it's part of. Even in isolation, a cell is a complete functional system. Paramecium is a perfect example of this. Despite being a single-celled organism, it eats, moves, reproduces, and responds to its environment, all within a single microscopic unit. This highlights a key point. Cells are not just building blocks. They are the smallest units of life capable of independent function. Let's also consider scale. Cells are microscopic, but they form the basis of all living systems. Trillions of cells make up the human body. Each one performs specific functions based on its type: skin cells, muscle cells, neurons, blood cells. They communicate through chemical signals and electrical impulses. They divide and replace themselves, constantly renewing the tissues they belong to. Even more impressively, all these cells come from a single fertilized egg, a single cell that is divided and differentiated into all the specialized forms we see. In plants, cells give rise to leaves, stems, and roots. They form the tissues that carry water, support growth, and create food through photosynthesis. In protists, cells must do everything on their own: gather nutrients, manage water balance, reproduce, and survive. Despite these different roles, the basic cellular mechanisms remain largely the same. DNA is universal. Ribosomes are universal. The need for ATP is universal. This is why cells are often called the universal unit of life. And it's also why studying them, even something as simple as a cheek cell or a sliver of onion skin, provides insights into life itself. This isn't just about memorizing organelles or recognizing shapes under a microscope. It's about understanding the logic of biology, the way structure and function come together to create living systems. It's about recognizing that something as small as a ribosome or a vesicle can have consequences for an entire organism, and appreciating that even the most basic cell is the product of billions of years of evolution, a highly optimized system shaped by time and adaptation. Noticing how the same parts, used in slightly different ways, can give rise to the vast diversity of life we see. So, whether you're looking at Elodea streaming chloroplasts, or a paramecium swimming in a droplet, or your own skin under magnification, you're seeing a common thread, the thread that ties all life together, the cell. And that brings our exploration to a close. Thanks for joining this quiet journey inside the smallest rooms of life. We've seen structure, function, diversity, and unity, all hidden in plain sight and all within the cell.